So last time we started talking about carbon NMR, right? What's the big difference between carbon NMR and proton NMR? Besides that, but that's true. Okay, well, typically we see all singlet peaks because of the way we run the experiment. What else? What about the scale? It's a much bigger scale, right? So proton NMR is from about 0 to 10/12, and carbon NMR goes from about 0 to 200/220. So a much larger scale to deal with, right? So having a much larger scale means that things are going to be spaced out quite a bit more. And what are these letters above? S, D, T, Q? All right, so that means that that carbon is a singlet, meaning how many hydrogens are attached to it? 0. If it's a doublet, how many hydrogens are attached to it? All right, and a triplet? Quartet? Very good. So we can see that we have at least how many unique environments? 1, 2, 3, 4, 5, 6, right? So can we eliminate anything based on that?
We only have 5.
Well, if I'm looking at it, I see 1, 2, 3, 4, 5, 6, 7. Right, because there's symmetry. 1, 2, 3, 4, 5, 6, 7. 7 unique carbons. So we can eliminate B. Let's see, we would have 1, 2, 3, 4, 5, 6, 7. Yes? So we can eliminate B and D. So we can't base it on that, can we? Notice down here I have 2 singlets, meaning that there is 2 carbon atoms that have no hydrogens attached to it. Is that true for C? We only have 1. So by process of elimination, we know it's got to be A, right? And if we look at it, it makes perfect sense, right? So we have these hydrogens on this carbon, right, that's attached to an oxygen, so it's going to be a little further downfield. There's 3 hydrogens on it, so it's a quartet. We have this carbon, right, which is attached to an oxygen. It's also attached to a benzene ring, so it's a little further downfield. And it's going to be a triplet because it has 2 hydrogens. These 2 are going to be singlets because there's no hydrogens to them, and then we're going to have a doublet and a doublet, and that's what we have. So we can put together the structure pretty easily when given the choices. And we were able to eliminate half of them based on looking at the unique number of carbons, which is kind of nice, right? It's 2 singlets. So 1, 2, 3, 4, 5, 6, 7, 8, 9 unique environments. This is TMS, and that's CDCl3, okay, that comes around 77. So we're going to ignore that as well.
So we're going to have 2 that are not attached to any hydrogen.
Very good. It's B. All right. We've got 1, 2, 3, 4, 5, 6. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9, that matches up, right? That should be a singlet, and that one should be a singlet, right? And that one appears pretty far down, around 165-ish, right? And if you look through it, you'll find that that all matches up quite nicely with the structure, whereas the other structures don't match up well. Now, in lab this week, right, you're figuring out how to use both carbon NMR, proton NMR, and IR, all your spectroscopic techniques, right? So here we've got a carbon spectrum and we've got a proton spectrum. Which structure do you think is in agreement with those spectra? That's an S? Mm-hmm. What comes out about 200 to 220? Aldehydes and or? Carboxylic acids actually come up around 165, 170. Ketones. That single peak tells me what I need to know. You can eliminate A, C, and D based on that peak right there, because you know that's where aldehydes and ketones are going to come. This is an ester, that's a ketone, that's an ester, and that's an alcohol, right? You can eliminate, eliminate, eliminate. Now, if we look at the spectrum a little more carefully, right? Let's look at the proton spectrum first. These three hydrogens, where do they appear? That singlet. There's no neighbors, right? How about this one? Triplet. It's going to be a little further downfield, right? How about this one? What's this one going to be? That's right. It's going to be that multiplet. And this one's going to be that triplet, right? So we can piece it all together. But by knowing that this is where aldehydes and ketones come, I know that. All I got to do is look, and I can eliminate a good chunk of them. In fact, I can eliminate 3/4 of them. And if you can eliminate 3/4, and you've only got 4, right? You should get that one right. All right, so that's the NMR stuff that we're going to do. And again, we'll use it at various points this semester and next semester, okay, as a technique. But to get really, really good at this, you'll have to spend years doing it, okay? So I'm not going to give you molecules that require you to really be savvy at NMR interpretation. You'll get that with experience as you study more and more chemistry, if you study more and more chemistry. But another thing that I want to talk about is mass spectrometry. This is a technique that is near and dear to my heart. However, I am not going to hold you responsible for it on the exam. Okay, I just want to talk with you about it so that you understand what it is. Mass spectrometry is a very, very good technique and allows us to determine what's known as the mass to charge ratio of a molecule. And if that charge happens to be 1, and usually it is in these experiments, then we can determine the molecular weight of our compound. So it's a nice way to determine molecular weight of an unknown. It is extraordinarily sensitive. Okay, so what we see here is the mass to charge, and if the charge happens to be 1, in this case it is, then each one of these represent a molecular species with its actual molecular weight. So this is of ethyl benzene, which has a molecular weight of 106. You can see the peak here at 106. Why do I see a little peak here at 107? Thanks, Casey. Pardon? Isotopes. What isotope? Carbon-13. That's right. So you'll see a little bit of carbon-13 in your molecule. So you'll see isotope peaks. Okay? That also is a carbon-13 peak, right? But notice, this is the molecule with a +1 charge. They've removed an electron from it. The machine can do that pretty easily, right? But what are all these other peaks? These are called daughters. Okay, so we're putting in ethyl benzene, which is a pure molecule, and when we remove an electron from it, we get daughter peaks. Can anybody tell me why?
Because the electron's still floating around.
We've removed the electron completely. Where am I going to remove an electron from? Where are electrons at in a molecule?
The electron cloud.
Where?
On the outside of the nucleus.
Well, that's true. Pardon? In the bonds. That's exactly right. So if I remove an electron from a bond, what happens to the stability of my molecule? It goes out the window, right? And so it decomposes into things that are more stable, okay? So if I remove an electron from ethyl benzene, the most stable thing that can decompose into is what's known as the benzyl carbocation. Why is that more stable than this?
Because it's got, it's a tertiary.
Well, it's not tertiary. That's really kind of like a primary, but why is that more stable than this one? Because I can draw resonance structures for that cation. And what happens when we draw resonance structures? It increases stability. And it turns out that this pattern, which we call a fragmentation pattern, is unique for ethylbenzene. How many things in the world do you think have a molecular weight of 106? Just ethylbenzene? No. There's a number of things that have a molecular weight of 106. Okay, so just knowing that peak by itself is useless. But knowing that and everything that it decomposes into gives me a lot of structural information. Okay? And so mass spec is a very, very powerful technique. It's the second most desired technique in a laboratory. NMR is the number one. NMR is non-destructive. I get my sample back. Okay? Mass spec is destructive. It destroys the sample during the analysis because we're removing electrons from it and it decomposes. Okay? This technique is also very useful for forensic scientists. This is how they determine illicit drugs, for the most part, is by mass spec. Okay? So this is a structure of cocaine. Cocaine has a molecular weight of 303. And let's suppose that Danielle is a forensic scientist and she says that Joe Blow's vehicle was pulled over and it had cocaine residue in it. And she goes up to the judge on the day that they're doing the trial and she says, "Judge, definitely was cocaine that had a molecular weight of 303. You know what their attorney's going to say? The defense attorney's going to say? Not only does cocaine have a molecular weight of 303, but here's a list of 1,000 other compounds that have a molecular weight of 303. My client can't be guilty based on that alone. But being the good forensic scientist that she is, she's going to say, not only does it have a molecular weight of 303, but it has all the daughters of cocaine. Okay? And when we compared the spectrum that I got from Joe Blow's pickup truck, right, and compared it to the database, the National Institutes of Standards and Technology database, it matches up perfectly. Now you've sent Joe Blow to jail, right? But 303 by itself doesn't do this. Yes, ma'am.
Is daughters like children or like daughters?
No, daughters are kind of like children.
Okay.
This is the parent, and it decomposes into its daughters. Okay? What's the base peak? Now, that's the molecular ion. The base peak is the tallest peak, 82. Right? That's the most stable species, is the base peak. Okay?
So is each peak like it looks as representative of what the molecule would look like if it was removed from all the different possible bonds?
Mm-hmm. Yep. Now, not every possible disconnection occurs because some of them aren't stable. But 272, and I don't remember which one this is, but that is the loss of some groups to make daughters. Yep, absolutely.
How does everything break down into daughters?
Depends on the technique. The technique that I'm showing you, yes, you will see daughters. Okay. We have what are called soft ionization techniques, which is what we use a lot, and they only show the molecular ion usually, unless we turn it on to do fragmentation. Okay. The other cool thing about this technique is extraordinarily sensitive. This is the technique that they use to determine whether or not athletes have been using anabolic steroids. This is what they use to see if racehorses have been doped. Okay? Because you can literally detect a frog peeing in Lake Erie with this technique. I mean, it's that sensitive. Okay? You can detect that. All right? It's that sensitive. You can detect down to the picomolar levels. Okay? So Joe Blow 3 years ago had a bag of cocaine in his truck. There's still enough residue in there that they can detect it after that period of time. Okay? It's that sensitive. They can detect whether or not you smoked marijuana 3 months ago. They can detect because it's that sensitive. Okay? They can determine whether or not a racehorse has been given loperamide. Okay?
So don't let your friends bring mysterious baggies into your car.
I wouldn't. So, what is mass spectrometry? Mass spectrometry is the analysis of molecules and molecular fragments, their ions, by measuring the mass to charge information. The information that you essentially get is the mass-to-charge ratio and the intensity of those mass-to-charge ratios. But if you're clever, you can use that information to do a whole lot of stuff. How many different mass spec techniques are there? There are a lot, but the common ones are electron ionization, which gives us spectra that look like this, which is what the forensic scientists like to use. What we like to use in my laboratory what are known as electrospray and MALDI. We won't talk about those today, though. Okay? They're soft ionization techniques, and they don't fragment the molecule very, very much. And you should be asking yourself, why should you care about mass spectrometry? Well, many of you said you want to go to medical school. If you do research in medical school where you're trying to figure out how a drug works, you're going to be using mass spectrometry because it's sensitive. Okay? You can detect molecules very, very easily in urine, feces, breath, analyzers, the whole 9 yards. Okay? You can detect those kinds of things. So my wife was actually in the pharmacology department at Vanderbilt when we were there. That's all she did most of the time was mass spectrometry on rat parts where they were trying to see what happened with the drug that they were administering. Okay? But they used it because they needed to detect at the extremely low levels where these metabolites were. Forensics, again, busting the bad guys. Environmental science, if you're interested in the environment, you go out and you want to detect pollution in waters and air and other things, mass spec is the technique to use most of the time. Biology uses mass spec quite a bit, and you should care about it because of the money. If you have skills in mass spectrometry, you will find a job. During the economic recession and big downturn, every friend that I had from Vanderbilt who had substantial mass spectrometry experience was hired during that period for no less than 6 figures. Okay? Pharmaceutical companies want you. Environmental companies want you. Government agencies want you. You can use it to detect explosives. You can use it to detect nerve agents. You can use it to detect all kinds of stuff. Each and every one of my friends makes more money than I do today because they have more experience in this than I do. Okay? And that's pretty cool. So you can make a lot of money if you have these skill sets. Okay? Now the way that this works, at least for the electron ionization, your sample comes in, you've got an electron beam. The electron beam kicks an electron out of the molecule by using the wave functions of the electrons. Those then get accelerated in a high vacuum through a field. In this case, they're showing a magnetic field. And then that allows the ions to be separated based on their mass-to-charge ratio. Okay, heavier ones go to one side, lighter ones go to the other, and a computer can detect them. Okay, and then once we have the spectrum, we can interpret it. So it's pretty straightforward system to use. So this is kind of like what an electron ionization source would look like. Don't use that word impact. It's actually a misnomer. Okay, but it's what's on the figure. Your molecules come in, they're neutral. The electron beam then removes an electron from your molecule, and they get accelerated to a negatively charged electrode that has a hole in it. And so we know that positive things are attracted to negative things, right? So these are accelerated towards that negative electrode, but there's a hole in it. So what happens? Some of them go right through it. Not all of them. A very small percentage actually go through it. But some of them go through, and then they go to the analyzer where we can separate them based on their mass-to-charge ratio and get that spectrum that we look at. Okay? Which is kind of cool. And on the way that they get there, they break down into their daughters. Okay? And so we can determine the mass-to-charge ratio pretty easily. All right. That's all I really want you to know about mass spec. We have 2 mass specs, not in my personal lab, but in the laboratories available here on campus. They're not huge. They're pretty expensive. So the ESI system that I like to use cost us almost $300,000, and the MALDI system that the polymer people like to use cost us about $250,000. It's a small drop in the bucket. So what I want you to do in the time that we have remaining, I want you to get with your friends, and I want you to interpret as best you can the spectrum for 2-octanol. And put your name on it because I'm going to have you turn it in for your inverted lecture. So we got about 10 minutes. So I would strongly recommend that you draw the structure of 2-octanol on there and use that as you interpret.